you measure the genetic diversity, and there are many ways of measuring genetic diversity. I'm not going to get into technicalities now. On the y-axis is a measure of genetic diversity, and as you can see that the measure of genetic diversity in Africa is the largest, followed by genetic diversity in Asia, and then the other parts of the world, Europe, Australia, etc. They are far smaller than either Europe or Asia. So the inference is that we evolved in Africa, and however we came to Asia, or we started living in Asia, we also have a very large amount of genetic diversity among population groups in Asia, which essentially means that those populations are also fairly old compared to other populations in Europe and other such places. If you do it, if you compare DNA sequences also, essentially it's the same. African populations, the diversity of African populations are on the are higher than populations in other parts of the world. So the inference from all of these genetic diversity studies is that we evolved in Africa, but we evolved in Africa and then did what is the question. I'll produce another piece of evidence that will actually build the story to what did we do after we evolved in Africa. So if you look at these numbers or these symbols, the color-coded rectangles and the labels on the top, these are signatures. These are genomic signatures. Essentially, it's a combination of nucleotides. It's a combination of alphabets, specific combinations of alphabets. And if you get specific combinations of alphabets, just as by comparing DNA sequences, we can understand, or we can statistically infer which DNA sequence is older than which other DNA sequences. Similarly, we can date these different kinds of signatures. A very unimaginative way of naming the signatures, A, B, C, D, etc. And these are color-coded, and the ones that are on the left-hand side are the ones that are older because they belong to the older branches of this phylogenetic tree of genetic signatures. The ones to the right are the ones that have evolved after those signatures have evolved, or from those signatures. Now, if you look at human populations resident all over the world, and if we ask ourselves, what kind of signatures do we get in these different regions, this is what the picture pans out to be. What we see is that it's slightly complicated. Each pie diagram actually is relative frequencies of these various kinds of signatures. It's color coded, so it's easy to read. I won't let you, take you through all of the colors and everything in detail. But what suffices is that if you look at Africa, you find that these are the signatures that are the oldest signatures are in Africa. If you look at the New World, which was populated because humankind crossed the Bering Strait between 15 and 20,000 years ago and went and peopled the New World, which is the Americas, you find that most of the newer signatures are in the New World. You don't find the older signatures. So, the Middle East, India, and all of these places comprise a diversity of signatures that all belong to the Middle Ages. The inference is that humankind evolved in Africa, and we moved out of Africa to essentially populate Europe, India, Middle East, etc., and then moved on to populate the New World, and therefore the newest kinds of signatures are what's found here. These are Y chromosomal signatures, and these are paternally transmitted signatures, so that's what it is. So using Y chromosomal genetic signatures, it's consistent with the fact that, or with the evidence that humans evolved in Africa, and it builds up the story that we probably moved out of Africa and started peopling other parts of the world. There is also extranuclear DNA that's outside of the nucleus. It's called mitochondrial DNA, which is a short piece of DNA, but even in that short piece, we find signatures. And if you look at maternal DNA signatures, again, these are from left to right. The left one signatures are the oldest, the right signatures are the newest. They are color-coded, and you find exactly the same feature. The oldest ones are in Africa, very diverse in the Middle East, India, and these kinds of places, Asia and Europe mostly. And in the New World, you find the newest signatures, predominant in the New World. So this is again consistent with the theory that humans evolved in Africa, moved out of Africa. We first, went and peopled Asia and other such places, and then moved on to people the New World. So this is what we know of modern humans. Modern humans meaning Homo sapiens, and this has been done using DNA sequences from contemporary human population. Like I said, that modern humans arose in Africa. When did the modern humans arise in Africa? We can again look at the DNA sequences, and they date back to about 150,000 years ago. The DNA sequences that belong to the modern humans can be dated to about 150,000 years ago, give or take 20, 30,000 years, but about that time. So we evolved in Africa about 150,000 years ago, and there are again the 𝐒𝐞𝐦𝐬 family, etc. They identified these skulls, and they have been also dated to about 130,000 years. Mine. So we look at Africa, and we started moving out of Africa, and these are the various migration trails that we followed. And by and large, you don't have to concentrate on this. By and large is what I've already told you. We populated Asia and Europe, and then moved on to populate the New World, which are the Americas. When we came out of Africa, did we meet any of our relatives? Did we meet any cousins that looked like us or was close to us? The answer is yes. We actually met several other species, and we were convinced that we were not alone. So we came out of Africa and met species like this. Many of them were, of course, Neanderthals, because Neanderthals did exist at that time. And if we predominantly there were two different kinds of cousins that we met, among others. The others I'm not getting into any detail, because they were very small in number, and they became extinct at some points of time, whose DNA sequences have not been possible to identify and to sequence. But I will talk about these the other kind of species as well. So we met two other species of hominids, of our cousins, in the Eurasian landmass. One is predominantly the Neanderthals, so we met the Neanderthals, and the other is called the Denisovans. And the Denisova is a cave in Siberia where a small finger bone was found, and that finger bone provided us with some DNA which could be sequenced. And when you compared the sequence of the finger bone, compared it to the Neanderthal DNA sequence, it was pretty convincing evidence that the Denisovans and the Neanderthals were two separate species. So these are the two major species that we met once we came out of Africa. And like I said, that the Neanderthals existed for almost 500,000 years in Europe and Western Asia, but they disappeared about between 30 and 40,000 years ago. And the questions that remain, the question that confronted many evolutionary biologists before Svante Pääbo was able to solve the puzzle, is that their history and relationship, there meaning the Neanderthals and the Denisovans, their history and relationship to modern humans, in other words, the relationship between the archaic humans and the modern humans, were unclear. They became extinct, the Neanderthals became extinct, and therefore the dominant proposal was that we exterminated them because we were good tool users. Incidentally, I must also mention that Neanderthals knew how to make stone tools. Yet the dominant theory was that we exterminated them. And the second theory that also prevailed is that Neanderthals are not direct ancestors of the present-day Europeans, and I'll explain this in a few minutes as I go along. That was another dominant theme. Both were actually proven to be false, and the main team that proved this was led by Svante Pääbo. So here is Svante Pääbo, and he's of course gotten the Nobel Prize for his discoveries concerning the genomes of extinct hominins and human evolution. So he was able to, like I said, that he was able to extract DNA from these fossil bones, and was able to actually sequence that DNA, but it was not an easy chore. It's easy to sequence, take a sample of my blood, isolate DNA and sequence that. That's become easy. but because of various other reasons, it wasn't easy to isolate DNA and to be able to sequence DNA from these archaic humans. The first one that he encountered, or he was successful in sequencing, came from this cave in Croatia, which is near this is Croatia, near the Adriatic, and this is called the Vindia cave. This particular cave provided a fossil specimen that he was able to again isolate DNA from, and these are the bone fragments. It was published in Science, cover of Science. They provided some bone fragments. The bone fragments were about 40,000 years old. And they were able to, Svante and his team were able to isolate DNA from that bone to generate a draft sequence. Why is it called a draft sequence? Because it was still incomplete. It was only done in bits and pieces and not the complete DNA sequence of the Neanderthal. Why was it not complete? Why is it so difficult? Some of which I have alluded to. First of all, it's not very well preserved. It's exposed to the vagaries of the weather, vagaries of sunlight, vagaries of other kinds of pathogens and other kinds of microbes that infest on these carcasses. So the DNA, because of all of these exposures, actually gets hugely fragmented. And once it gets very fragmented, it becomes very, very difficult to sequence those small pieces of DNA. Won't get into more details of this. There's also an false overrepresentation of some alphabets, and that false overrepresentation is because of certain chemical changes that take place. Again, something that I will not get into. The other is that there are certain blockers because of this vagaries of exposure, etc. There are some DNA modifications that take place, and because of these modifications, sequencing becomes almost an impossibility. So this prevents amplification and sequencing. By the way, the DNA that we isolate from a fossil or even from us is so small that we can't directly sequence it. We need larger amounts of that DNA. And to be able to manufacture larger amounts, it's done artificially by a process called polymerase chain reaction. And the person who actually discovered or invented polymerase chain reaction also got a Nobel Prize. His name is 𝐸. But these kinds of blocking lesions actually prevent the amplification of DNA. And if you cannot amplify DNA, it becomes impossible to actually sequence the DNA. Another major reason is miscoding. There is false incorporation of certain amplifications, certain nucleotides during amplification, again because of chemical changes. So all of these problems had to be overcome before you could sequence the first Neanderthal DNA, and it's really, really an enormous task that he overcame. There's contamination with exogenous DNA, like I already mentioned. That the exogenous DNA is like the bacterial DNA and the fungal DNA, and you have to eliminate all of that. Otherwise, you would be only producing sequences of bacterial or fungal DNA. But that's really not what you want to do. There is yet another big problem, which is called racemization. When amino acids, live within us, when we have amino acids which are encoded by the DNA, and these are available in two forms, left-handed form and right-handed form. When they're outside of the body, not doing any physiological processes, there is conversion from one form to another. Ultimately, if you leave it for a long time, you get 50-50 of two kinds, left-handed and right-handed. But because of certain chemical processes, if you take ancient DNA and if you try and identify, because of the changes in the amino acid, if you try and sequence, then you get false sequences because those kinds of chemical changes have already taken place, and racemization is a major problem in sequencing of the ancient DNA. What does it boil down to? It boils down to the fact that even if we aspire to sequence the DNA, it becomes an enormously difficult chore to be able to isolate DNA, to be able to clean up the DNA, to be able to sequence the DNA. All of these needed to be overcome, and Svante Pääbo painstakingly, with a great amount of patience for over several decades, actually did this and was able to sequence ancient DNA. The other thing that he did, having been able to successfully sequence DNA, he was also able to find certain specific points in genomes that don't belong to us. Essentially, he was able to identify certain genomic signatures of the Neanderthals that do not belong to us, and because he was able to identify those specific signatures, he could also try and answer the question, did we get any portion of our DNA from the Neanderthals? For that, he needed to identify these specific spots in the Neanderthal DNA which did not match with the humans, but later, as we'll see, they were contributed to the human DNA, and that's how we know that there was. Well, I won't give away the story right now. Okay, so those are that Neanderthal DNA, and those red spots belong to the Neanderthals. We also find red spots in some of the humans. These are all human pieces, the blue ones, and these are variant positions in the human DNA. But there are a few humans who also have these specific positions that actually belong to the Neanderthals, and they also are available in some human DNA. Why is it so, and what's the interpretation of this? We'll come to in a minute. So he was able to produce the draft sequence of the Neanderthal genome, and that was in 2010. That was the first draft sequence of the Neanderthal genome that was he was able to produce. He also was able to compare the Neanderthal genome with the human genome and obtain some very useful inferences. Number one, Neanderthals are closer to non-Africans than to Africans. And the reason is because the Neanderthals, the predecessors, ancestors of the Neanderthals, left Africa about 500,000 years ago, and they were mostly in Europe and Asia. And therefore, they are more close to non-Africans than to Africans. Why did this happen? That was only a piece of observation, that their genomes are closer to non-African genomes than to African genomes. One reason is because they left Africa a long time ago, but why are they closer to non-Africans than to Africans? We never know for sure. So that needed an explanation, and we'll come to that explanation very soon. The direction of gene flow, so there was admixture, there is interbreeding, there is a direction of gene flow, and he was able to show, using those specific red spots in the Neanderthal genome that were available in some humans, but not the other way around. You couldn't get the human-specific DNA spots in the Neanderthal genome, meaning you couldn't get the blue spots in the Neanderthal genome, but you did get the red Neanderthal spots in the human genome. That essentially meant that the direction of gene flow was from the Neanderthal to the human, but not the other way around. The third evidence, the third inference that was that he drew in this particular paper is that segments of Neanderthal ancestry in the non-African genomes could be identified. And the third and the last thing that he did is that he compared this one draft genome, and this was again a little premature because using a sample of size 1, you are comparing with by then several thousand human genomes that are already available. But then he used that data in order to estimate the amount of Neanderthal ancestry in the humans, because Neanderthal genomes had introgressed into the humans because of interbreeding, and the extent of Neanderthal admixture was between 1% and 4%. It's very interesting that even with the sample size 1, when he drew this inference, that inference has held up even after we have sequenced more than 1,000 Neanderthal genomes. So that's very, very interesting. Essentially, DNA evidence is very hard evidence, and that's the reason why the estimates are fairly stable, even if it's fragmentary. The complete genome sequence was then done in 2014, 4 years later. It took them, again, a lot of doing in order to get a complete genome sequence from the draft sequence, and then there was no looking back. The methods have been all perfected. We know how to clean up, we know how to amplify, we know how to sequence ancient genomes. And all of that credit goes to primarily to Svante Pääbo. At some point of time, when you're trying to do this and you repeatedly face failures, you become frustrated. And Svante expressed frustration. And I'll read out 2 such sentences. He said that we'll never be able to sequence the nuclear genome of a Neanderthal. It's too degraded. I've already said that these DNA pieces, when exposed to the vagaries of the weather and other such kinds of things, they get degraded, they get fragmented, they get infested by bacteria, viruses, fungi, and so on and so forth. Also, the amount of DNA that you can extract from these bone fragments and skull fragments is just too little. There's too little DNA there, it can't be done. So this is Svante Pääbo, this year's Nobel laureate, who actually made these statements out of frustration after he tried for years to identify or isolate ancient DNA. Then he says something that we must all learn from. He says, of course, you should never say these things, never say things like that, particularly in biology, because you'll often be overtaken by technical developments. And indeed, he was one of the persons who actually was fundamental to development of these kinds of technical developments. So he was a prime mover of these technical developments. And this is in 2015, when he was giving a named lecture at the Cold Spring Harbor Laboratory. And this is a year after the complete genome sequence of the Neanderthals was published. How many ancient genomes have we, how many Neanderthal genomes have we actually sequenced so far? Over 8,000. Where did they come from? As you can see, they come primarily from Asia, primarily from Europe, some from North America, South America as well, but almost nothing from places like India, the Middle East, etc. And the reason why it mostly comes from Europe is because Europe is like a cold storage, right? So if you put muscle in the cold storage, the muscle meat in the cold storage, it survives for a very long time. It doesn't get degraded. But in places like India, where many people have been trying to isolate ancient DNA from whatever fossil remains that we have, it's been almost impossible, primarily because of degradation, vagaries of the weather, and also contamination with other microbes. So that's the Denisovan's cave that I showed you, and a little finger bone was found. It was sequenced, and it was found that this sequence does not match with the Neanderthals. It could not even have been derived from the Neanderthals, doesn't match with the humans, could not have been derived from the humans, and that then it was declared as a separate species, and that species has been named as Denisovan's after the name of the cave, which is Denisova. And the group actually identified the genetic history of another archaic hominin from this particular cave in Siberia. They have an evolutionary history which is distinct from the Neanderthals and the modern humans. I've said most of these things that there was a common ancestor and they lived a long time ago, etc. They moved to Eurasia. Modern humans arose in Africa about 150,000 years ago. We started moving out of Africa about 100,000 years ago. Again, probably pressure on natural resources and our inquisitiveness to explore. We came out of Africa. We went places. We went to the Middle East. One of the first places that we came to after we moved out of Africa is India. So we have a very high genetic diversity among populations of India. We reached the Near East sometime about 50 or 60,000 years ago, and we started to mix with the Neanderthals, predominantly somewhere in the Middle East, before continuing through Europe and other parts of Asia. We also mixed with the Denisovans in Southeast Asia and continued on to the Pacific. So we do have some admixture with the Denisovans in the Pacific, and as a matter of fact, the extent of admixture with population groups of Pacific with the Denisovans is higher than those in Europe, for example. But then the Neanderthals and Denisovans became extinct. But they live on because, like I said, that specific signatures in the Neanderthal genomes have been identified. Similarly, specific signatures in the Denisovan genome were identified. And then these three species lived in the same geographical region. One of the things that they probably did was to interbreed, as a result of which Neanderthal genomes came into human genomes, because we do find Neanderthal genetic signatures in the human genome. The extent of admixture was about 1 to 2% of modern humans of non-African ancestry, and about 7% of people in Melanesia. So as you can see that these archaic hominins did contribute their DNA to us, and the extent of DNA that they have contributed varies from one region of the world to another region of the world. And the variation is quite large, from 1 to 2% to about 7% in the people of Melanesia. The Okay, so I'm not going to read this. I'm also not going to explain this. This is also very interesting. But what I want to point out is that 25% of these signatures, they're also called haplotypes. 25% of these signatures that are non-specific, that have been identified, actually do not identified in the humans, actually do not match either with the Denisovans or with the Neanderthals. So they probably are derived from an as yet unknown archaic hominin, which essentially means that there were other species of humans with whom we were interbreeding, but those fossils have not been found. Their DNA sequences have not been sequenced, and therefore we do not know how many more other species that we mated with. We do have evidence about the Neanderthals and the Denisovans. We have been interested in human evolution for a very long period of time, and we've been working in among population groups of India and elsewhere. So I will just provide you with two interesting observations that we made from our own data. We were working in the Andaman and Nicobar Islands, specifically among the Jarwas and the Onges. These are two tribal populations in the Andaman and Nicobar Islands. Look, very African, very dark, frizzly hair, and so on and so forth. Morphologically very African, and so they always had a lot of interest about why they look so different from us. Essentially, we got some blood samples from them, isolated DNA, and identified that in the DNA sequences there were some small pieces that actually didn't belong to us, didn't belong to the modern humans. It didn't even belong to the Neanderthals or the Denisovans. These yellow pieces didn't even belong to the colors have changed. Anyways, so these yellow pieces did not belong to the Neanderthals and the Denisovans, but they dated back to about the same time when the Neanderthals and Denisovans actually roamed around in Europe and parts of Asia. So what we said is that these pieces don't belong to any of the three species, us, Neanderthals and Denisovans. They must have come from another unknown hominin. And we actually published this soon after the first Neanderthal complete sequence was published. And I'm going to explain the figure, but that was published by us a few years ago. We then, around the same time, there are other people who are also working among tribals of Melanesia. They also found something very similar. They also found DNA sequences among some tribal populations of Melanesia that from which they concluded that these early Melanesians actually interbred with the mysterious hominid. The mysterious hominid is an unknown hominid whose fossil remains have not been found. We also went ahead and looked at various other populations in Asia in a collaborative mode with other people. Most of these, and then we published our study in together collaboratively. Most of the samples in that study came from India, and as you can see that they came from India, from various parts of India and also Pakistan. We collaborated with groups in Pakistan. But what is it that I want to tell you today? I won't get into the details of the population structure of Asia or of India. What I do want to tell you is that we estimated the extent of Neanderthal and Denisovan admixture among population groups of India. So these are various population groups of India. The names of the populations have been coded, but doesn't really matter. As you can see that there are, overall it varies from about 1.9% to 2.49, 2.5%. So about 2% to 2.5% is the extent of Neanderthal genomes that we carry amongst ourselves, population groups of India. How much of Denisovan admixture had happened? Much less. The amount of Denisovan admixture in our genomes, or Denisovan genomes in our own genomes in India is 0.8, about 0.1% to 0.4%. So Denisovan admixture was much smaller than Neanderthal admixture. If you go to go to the Pacific, it's just the other way around. Neanderthal admixture was much smaller than the extent of Denisovan admixture in populations of the Pacific. So there are regional variations, and these regional variations have happened depending on where the Neanderthals or Denisovans were more prominent, numerically more prominent. I want to end my talk with one question. Again, the question was lurking at the back of our own minds, but it was Svante Pääbo and his group who answered this question. We talked about, we said that there is no, that there's mating between Neanderthals and humans. There was mating between Denisovans and humans. We found Denisovan DNA in the human DNA. But we didn't find human DNA in Denisovan DNA. How did that happen? So it was initially there were lots of speculations, but now again the way that we do it is that we generate data and then we have competing models. We try and identify which model fits the data best, or the data support which model the strongest. So one of the several models, and I'm not going to narrate all of the models, of the several models that were propounded, one model was the following: that the Neanderthals and the Denisovans, Neanderthals and the humans mated, but the offspring all went with the humans, did not go back to the Neanderthals. So if they did go back to the Neanderthals, then there would be matings between Neanderthals, and some of the human genomes would have gotten into the Neanderthal genomes as well. But since the offspring, the model states that the offspring went with the humans, then there would be no possibility of human DNA going back to the Neanderthals. And that particular model was tested multiple times. And as a matter of fact, that's the most parsimonious model that we have as of now. That after the interbreeding, after they procreated and offspring were produced, the offspring went with the humans and did not go back to the Neanderthals. The same thing happened with the Denisovans. But the question also remained, did the Denisovans and the Neanderthals interbreed? It took a long time to solve that mystery. There was not sufficient data. The major impediment was because not many fossil remains of Denisovans have been found. As a matter of fact, as of today, I think the third specimen is being sequenced. There are only 2 available Denisovan sequences, but over 8,000 Neanderthal sequences. This is what was found. And again, the ones with the blue shirt are the Neanderthals. The ones with the red shirt are the Denisovans, and the ones with the yellow shirt are the modern, ancient modern humans. I'm not going to take you through the entire thing. I'm only going to point out one particular individual here. This individual is 50% Neanderthal, 50% blue, 50% red, which essentially means that this individual is 50% Neanderthal and 50% Denisovan. That can only happen if one is a father and one's a mother, and the DNA sequences from the parents are contributed to the offspring. So this can happen if this individual is an offspring of a mating between Denisovan and the Neanderthal. As a matter of fact, they did the analysis of their DNA sequences, and that's the title of their paper: The Genome of the Offspring of a Neanderthal Mother and a Denisovan Father. So how do you know which is the mother and which is the father? Of course, there are paternally inherited the Y chromosome and the X chromosome and the mitochondrial genome. They analyzed all of that and were able to identify who was the father and who was the mother. And they identified that the Neanderthal was the mother and the Denisovan was the father. And so we now know that there was free intermixing between all of these 3 species: Neanderthal, Denisovans, and modern humans. So that was an extremely important landmark inference in evolutionary biology of humans, and it could not have been done without the great insights and the great contributions of this one single man and his team, called Svante Pääbo and his team. Later, there's another person who's shot into prominence. His name is David Frisch. He's from the MIT Harvard school. Want to end this by saying that, I said that, and his Nobel citation also states that it is primarily because of human evolution, but he's also contributed to an understanding of susceptibility and resistance to diseases in humans in relation to our interbreeding with Neanderthals and Denisovans. He's given multiple examples of this, and I give you one example that's close to us as of now, which is COVID-19. Many of us actually have gotten infected, came down with COVID-19, and have survived. Many of us did not actually get severe COVID-19, and it was mild, and we have survived. Many people got severe COVID-19. Some survived, some succumbed and passed away. So there are now studies that have been done looking at people who are infected and people were infected and nearly asymptomatic or mild symptoms versus infected people infected with SARS-CoV-2 and had serious symptoms of COVID-19 or had severe COVID-19. And genomic studies were done by comparing these two groups of individuals. It's called a case control study usually, where you try to identify whether there are genetic factors involved in predisposition to severe COVID-19. And again, this is, I'm not going to be able to tell you the details of the next picture. This is what they had found, and this is I'm giving you the punchline first. And this is from Svante Pääbo's group. The major genetic risk factor for severe COVID-19 is inherited from the Neanderthals. I'm not going to explain this. All that I'm going to tell you is that these are various loci, these are various points on the chromosomes that we have. And the y-axis is essentially a measure of association with severe COVID-19. And as you can see that there is one spike here, which essentially means that on this particular chromosome, there is a specific position that shows a great association with severe COVID-19. So that is the predisposing factor. So if you have that particular chromosomal, that particular sequence in your chromosome, then you are predisposed or susceptible to severe COVID-19 if you get infected with SARS-CoV-2. The question then came, where did this region come from? Can we identify the history of this genomic region? And they did painstaking work. Svante Pääbo and his team did painstaking work to identify that this region actually can be traced back to the Neanderthal. So the conclusion was that this particular region that actually provides us with susceptibility to severe COVID-19 was derived from the Neanderthal. He's given us plenty more examples that has to do with human health and disease in relation to our interbreeding with the Neanderthals and Denisovans that actually resulted in various kinds of genomic portions from the Neanderthal or Denisovan genomes to come into us. The next question that many of us ask is that if this region was derived from the Neanderthals, and since the Neanderthals actually provided different kinds of admixture, different proportions of admixture to human genomes living in modern human genomes living in different places of the world, where are the most predisposed individuals? Which populations or which regions have the highest frequency of this genomic region? And this is unbelievable. It's very striking. The place that is most predisposed to severe COVID-19 is India, as you can see, different populations of India. And we now have data to show that this particular genomic region is enriched in many populations of India. The prevalence of this genomic region is very high in multiple populations of India. I gave this talk not so long ago in 𝐼𝐶𝐸R, as a matter of fact, day before yesterday, to students in 𝐼𝐶𝐸R, and one student said that, but sir, we don't have data from any other places. Yes, we don't have data. So all of this inferences, like I'm saying with a great amount of emphasis, that this particular genomic region is of a very high frequency in India. That's a good statement, but whether it is the highest among all parts of the world, we still do not know because data are still being gathered. But as you can see from this particular graph, that it is fairly high, that we are very highly predisposed to getting severe COVID-19 once we are infected with SARS-CoV-2. Let me explain the title of my talk. It must have become clear by now. The title of my talk is We Moved, We Embraced, We Absorbed. We moved out of Africa. I hope that I've been able to give you enough evidence, enough idea that we actually evolved in Africa, moved out of Africa. We met the Neanderthals, Denisovans, and other hominin species whose DNA we still have not been able to isolate and sequence, but we met our cousins. We embraced them, and not only did we embrace them, we interbred with them. So we have their DNA in us. And like I said, that the most parsimonious model is that we absorb them. Their children, after the children were born out of those matings, the children came with us and did not go back to the other parent. So we moved, we embraced, and we absorbed. Thank you very much. That's the story of human evolution.
was really an illuminating talk. There are many talks that we know about. And now, invite the audience to ask questions. Yeah.
The person who is asking a question is actually a long-time collaborator, and she herself has contributed to human evolution. And it's really nice that she could come to my talk. Thank you so much, sir. Thank you.
So I have a question. If we understand tradition, is this some kind of viral infection?
Well, there are multiple theories of that, of the extinction. One is that their numbers continue to dwindle because their children came with the humans. So there would be a demographic decline. That's one theory. We never know for sure. The second is, more recently, people have been able to identify around that time there was a severe climate change. So climate change has also been talked about. Whether there was a viral infection, I don't know, but that also I've read in places where they have said that it could have been because of a pathogen that exterminated them. But I find that very hard to believe because humans were also living with them, and a pathogen that could infect Neanderthals and exterminate them could also infect us and exterminate us. So why did that not happen? So I think this pathogen explanation or hypothesis is not a tenable hypothesis, but climate change and more importantly the demographic decline because children came with us is the more tenable ones for me.
India seems to have very different populations, like in terms of the demographics,
Sure. Sure.
I mean, how are they so different?
Oh, you're talking about the population structure of the modern humans living in India. Yeah, just the time scale is a little different to me. If we talk about that time scale, we had 4 different kinds of ancestors. Now we have DNA evidence and we have analyzed that data and we can trace back to 4 different ancestors, and the time scale is different. So we are not talking about Neanderthals and Denisovans. We are talking about much more recent ancestors. Probably 60,000, 70,000 years ago, and here we are talking about ancestors that lived about 1 million years ago. From the Neanderthals, yes, absolutely. Yeah, that only shows that there is continuity of evolution for sure. Yes, but that doesn't explain the current population structure. The current population structure is more complex.
Sir, how it became possible that our offspring is going to be the modern human and not with the Neanderthals or the dinosaurs? Is it possible to share all the offspring with this?
Yeah. So the reason why it may have happened, we'll never know that for sure. The reason why it may have happened is that we were also more intelligent. Compared to the Neanderthals, if you take brain size and so on and so forth, there are multiple ways to figure out who were better tool users, who were more intelligent and so on and so forth. So possibly because of that, but that's only a matter of speculation. Incidentally, this is also a model. If you come up with a better model that fits the data better, then that model will become more parsimonious. As of now, the most parsimonious model is that their children went with the human. This is not to rule out the existence of other possible models. How are we separating so what happens is that when a large number of these variants accumulate, after a certain time, separate species, meaning the distance between the DNA sequences is very large. So if you randomly draw two humans and you draw one human and one Neanderthal, the difference between the DNA sequences is so large that you wouldn't call them as members of the same species. That's all there is to it. When breeding stops is a separate issue. They still interbred, but they're called different species just because of the differences in the number of nucleotides.
Maybe a very basic, what sort of relation is there between the genetic diversity and the time? And it's linearly growth or non-linear?
A good question. So there's been a lot of research on this. If you look at different portions of the DNA, overall, if I was to make a statement, it's linear. So if you look at time versus the number of mutations accumulating, it's very linear. But there are portions of the genome where the accumulation of mutations is much more rapid and non-linear compared to the overall. So well, it more reflects to its non-bearing on natural selection. We can talk about that on the side. Yeah. So if certain regions of the genome are under pressure of natural selection, so you would not accumulate a lot of mutations there. They evolve linearly. But there are lots of non-coding regions in the human genome. Much of it is regulatory, and there also mutations don't accumulate so fast. But there are some regions where mutations accumulate very fast and in a non-linear fashion. But overall, it's linear.
So there's a question from live.
Okay.
There is a question: can DNA be linked to the human behavior? There should be some human characteristics that are acquired culturally, isn't so? That's the question.
Yeah. So I would say that not all human behavior is genetic. There is a lot of acquired human behavior, but some of it can be traced back to genes. Some aspects of human behavior can be traced back to genes.
Another is from YouTube. So, what is the reasons for the extinction of the Neanderthal and Denisovans?
So I just mentioned, I hope that I've answered that question. The main reason being that demographic decline, because the offspring went with the humans. That's the model right now. But there are also other competing theories such as climate change and other theories.
Okay, so another question is from Facebook Live. The question is, if Neanderthals and Denisovans were different species, how did they mate with archaic humans? And because, as per the commenters, he said that because the biological definition of the species builder is to produce fertile offspring, did the crossover of Neanderthal DNA happen only once?
Yeah, so that used to be the definition of a species when the offspring would be infertile. That's no longer the definition, and I just answered that question in relation to speciation just now. Beyond a certain number of accumulation of mutations, we would call them as different species. They do not necessarily have to produce infertile offspring. That's a more modern definition of a species.
Thank you. Any questions from the audience?
There are, I think, here there are some. I see some hands here. Yeah, just speak up. I can hear you. I'll repeat your question. Yeah. Hi.
So I wanted to ask whether if there is any effect of the genome or the Denisovan genome, the very infertility or the thing of the size of the Y chromosome.
I do not know that. I do not have an answer to this question, unfortunately. I'm sorry.
Another question from North Bengal, one of the live session is going there. So the question is why Pääbo sequenced the mitochondrial DNA in his experiments.
Why did he or did he not?
Why you guys used?
Why did he?
Yeah.
That's very natural, because DNA is within the nucleus, and extranuclear DNA is mitochondria, which is maternally inherited, passed on from the mother to all her offspring. There is an added interest to find out sex-specific admixtures and sex-specific movements. That's why we actually sequence the mitochondrial DNA. There's a great interest. I can spend another half an hour talking about the mitochondrial DNA.
Yeah. Any questions from audience?
There are some questions from here. Yeah, maybe come forward. Tahole subida hobe.
What does it mean by we?
We modern humans. What it means is modern humans, you and I.
So actually, I'm trying to say That does it mean human beings all over the world or specific region?
No. What it means is that when we evolved as modern humans in Africa, we came out of Africa, so we moved. We are, by saying we, I'm only mentioning about modern humans, the species, the modern humans as a whole, Homo sapiens as a whole.
Can we say beforehand which population is more prone to some particular disease, or do we need the genome of the new virus? As we did for COVID-19, that is, some particular population is susceptible to COVID-19. Can we say beforehand that
if you have identified a genomic region that predisposes you to a particular disease, yeah, then you can look at that particular region, the genomes of individuals, and figure out whether that population is susceptible to the disease.
We need the genome first.
You certainly do need the genome. Otherwise, if it's genetic, then you need to identify the genomic region to make an inference. Yes. You may not need to sequence the entire genome, by the way. That's what I meant. Yeah.
That integrity has taken place in practice study.
Yes.
My question is, could it be this way, we think this way, that offsprings born to Neanderthal mothers and offsprings born to Homo sapien mothers, they were genetically different, and that's why offspring belonging to Neanderthal mothers did not survive, and whereas the other way around, offspring born to mothers survive.
So I don't think we have related this to survival at all. What we related this to is that all offspring survived, but then offspring needs to go with a group, right? Now, whether they went with the fathers or with the mothers, we still don't know. That will require much more data. So it's not related to survival at all. It's to which particular group do you then, after your birth, associate yourself with. So it's not related to survival. But whether the offspring went with the father's group or with the mother's group, only time will tell. We don't have as much sex-specific data to be able to say that.
So, one question.
Come forward. Come forward. They'll give you the microphone.
Yeah. So, question.
Yeah.
On the live. So, about the COVID-19 susceptibility. So, does it mean that Neanderthals were immunologically weaker than the modern humans?
We don't know that. We actually don't know that whether COVID-19, the Neanderthals also succumb to COVID-19 or to SARS-CoV-2 infection. We would not know this. It's quite possible that they had other kinds of resistance factors that actually prevented susceptibility, or immunological factors. I don't know that. I mean, we don't have data on COVID-19 in Neanderthals. Until we have that, we can't make a claim on any of these questions.
So, was there any interspecific competition between like they competed for the survival, or
Don't know.
Who got extinct first?
No, these dates are very, very difficult to pin down, exact dates. Again, both of them 30 to 40,000 years ago. Whether there was interspecific competition, I would speculate yes, but no hard evidence.
There are two questions. Is this branch of knowledge medical anthropology? Another question I have, that the human types in Andaman and Nicobar are so varied. Is it only just one specific genomic pattern, or they have different genomic patterns?
No, there's a lot of diversity even in the tribal populations of Andaman and Nicobar Islands.
They look very dissimilar also.
They look very dissimilar as well, yes. With respect to your first question, I would rather refrain from categorizing this branch of knowledge to a particular discipline. I think it's very interdisciplinary. This is very statistical, it's very technological, it's very genomic, it's involves field work and all of this. So, meaning anthropology. So it's really a combination of various kinds of things. It's best not to try to classify this branch of knowledge into one category.
Okay, so another question is from live is that whether the DNA remains susceptible to the mutation after the death of the living specimen.
Oh, if you call changes in the DNA after the individual dies, those would not be called as mutations. They would be changes in the DNA, but not as a result of other kinds of biological processes that are usually responsible. I've mentioned about lots of different chemical changes that take place that leads to changes in the DNA, that leads to removal of lots of alphabets, etc. So you could call them as mutations, but that's not how a biologist would call them as mutations. They're just changes in the DNA.
So there is no correction process.
There's no correction process, of course not. It has to be physiologically active for the correction process to take place.
Okay.
As we find many data that users have larger brain modern humans, is it because they were learning more skills than us, or is it?
The Neanderthals had larger brain size than modern humans, but that's probably not true. No. The average brain size of Neanderthal was a few cubic centimeters smaller than ours.
So our cognitive capacity is larger.
It's not so easy to relate just the cubic centimeters of the brain to intelligence or cognition. It's not a straight line. It's not linear at all. Very difficult.
So another interesting question: why the Europeans are taller? That is the effect of Neanderthal gene. Percentage of the Neanderthal gene is higher there.
Percentage of Neanderthal gene is certainly higher there, but whether that segment that contributes to tallness or to height came from the Neanderthals hasn't been figured out yet. I think people have that particular hypothesis and they are looking. So we may actually find an answer soon.
And next question is, why is the Africa that the migration started from? Is there any biological reason or the geographic?
Why is Africa the cradle of evolution? Period. Nobody knows. You look at non-human species, lots of species actually evolved in Africa and again moved out of Africa. Why that happened, why Africa is so heavenly, one never knows. I don't know.
If the Neanderthal gene is susceptible to the SARS-CoV-2 virus.
Oh yeah, okay, sorry.
If the Neanderthal gene is susceptible to the SARS-CoV-2 virus, can we synthesize the virus to make our host species go extinct? Like we can.
Theoretically, yes. Yes, it's possible. But how will you infect people? You'll be put behind the bars before you can infect people. So I think it's possible. Honestly, I think it's possible. It's possible to synthesize pieces of DNA that will infect. Yes, absolutely.
If there is no question, then any more questions from audience? Okay, so now it is our turn to thank Professor.
Oh, thank you so much. Great pleasure. Thank you for coming.
As a mark of our gratitude, we would like to present him some books. I request Professor also to. We are actually giving him a few copies of Breakthrough and the Prophecy that we published, the book Science in Ancient India, and also A Brief History of Science. These are the books that we have. Now, it is my pleasure to thank all the students and faculty members who have come here to attend this lecture, not only right here, but also on the net. There is a very large number of people who attended this talk. I would like to thank them. There are two universities in which students and faculty members gathered in halls, and the talk was projected on screen. So it has been done that way in many universities.
So I would like to thank them for that initiative also. And I would like to thank the faculty members of this university for having helped us in getting the hall. Yes, and the staff who have helped us in organizing its thing. Thank you very much.